Welcome to Class Time, your daily classroom for CSEC and CAPE subjects. I am Joanna George Johnson, your CAPE biology teacher for today. Last week we spoke about photosynthesis. Today we are going to continue that discussion. Okay, so here we go. Last week, from last week's class, you should be familiar with this diagram that's on the board that is outlining the first stage of photosynthesis, which is the light-dependent stage of photosynthesis, which means this is a stage of photosynthesis that requires light. So we are going to just break down each step for you as we go through very quickly about what we did last week. First thing to happen is our photosystem, and we start with photosystem 2, is that our photosystems are hit with light coming from the sun preferably, because the sun will have enough energy to fire up or energize our molecules so that they can get to the second stage, or the second stage where the reaction center, which is a specialized chlorophyll molecule, gets energized enough so 2 electrons are elevated to a higher energy level so that it can be captured by a primary electron acceptor. Now we know that now the P680 requires some electrons because it has just lost its electrons, but it cannot get the ones that it just lost because those are too hot for it to handle. So therefore it has to get its electrons from the water molecule that will be split to produce these 2 electrons. Now, the electrons that left the PS2 is carried along a series of electron transport, and then it eventually going towards the PS1. Then what happens next is that during that transportation of the 2 electrons, the ATP molecules will be made from the energy that is released as the electrons make their way towards PS1. Now, number 6 shows that PS1 is also energized by the light from the sun. Hopefully, it doesn't have to be the sun, but usually it is because most plants are outside. All right? And then after that, yes, P700, which is associated with PS1, has 2 electrons being elevated from it that goes and captured by the primary electron acceptor. And then we have where the chain of electron transport occurs again, allowing for the final electron acceptor, which is the NADP. It is going to be collecting those 2 electrons along with the hydrogen ions that were given off from the splitting of the water, and so therefore make it NADPH, which is, as you know, the last part of the this whole series of actions that happens in the light-dependent stage. All right? Now, this diagram sort of outlines the whole thing, but in context of the thylakoid membrane. Remember, the thylakoid membrane is where this is taking place. This is a part of the chloroplast. Now, if you remember clearly, is that the thylakoid membrane has with it, if I was to draw something representing the thylakoid membrane, the membrane is where all the proteins are on. But then inside here, all of this space inside here of the thylakoid membrane, it is just empty space, and this is where all of the hydrogen ions that are made from the splitting of the water molecule will be collected. So you end up with a whole bunch of hydrogen ions in the space, and they want to come out. And because they want to come out, they can't just come out like that because they are charged, because they're hydrogen ions. The plus sign tells you they are charged. So they can't just come through the membrane, just easily like that. They have to go through a particular door, which is called ATP synthase. ATP synthase is a specialized protein that's found on the membrane itself. And what happens is, as the hydrogen ions make their way out from inside of the stroma, not the stroma, but the thylakoid lumen, they will pass through this protein molecule, and as they do so, energy is given off for the formation of ATP. And if you remember clearly, these are the two substances that are made, important substances that are made by the light-dependent stage of photosynthesis, so we can go into the next stage of photosynthesis, which will be the major part of today's lesson, okay? So let us get right to it, all right? So the light-dependent stage of photosynthesis we are going to be doing today is called the Calvin cycle. We are going to outline it in some detail, and we are going to, hopefully, we appreciate the importance of the light-independent stage, how it is dependent on the light-dependent stage, which we just did, okay? Now, this part of photosynthesis does not require light. That does not mean it occurs in the dark. All it means that if there is no light, it could happen. However, because it relies on the things, on the production of what goes on in the light-dependent stage, it does not occur in the night, in real life, okay? It also occurs in the stroma of the chloroplast. Earlier, for the light-dependent stage, that was occurring in the thylakoid membrane itself. This time, it occurs in the stroma. This is a stage where the sugar is made, and that's the ultimate goal of photosynthesis, as we know. The purpose of photosynthesis is to make sugar or to make food for plants. That is how plants make their food. So therefore, this is where, what the purpose is of photosynthesis occurs. And it utilizes the products of the first stage of photosynthesis. We are going to show that as well. And you should know by now the first stage of photosynthesis, those products are ATP and NADPH. Depending on which book you are using or which textbooks you may be pursuing or what online resource you are going to use, you may see reduced NADP instead of NADPH. And I'll explain later about why we call that reduce. Anyway, just to go back to the chloroplast so you have an idea where all of this is happening. All of this is happening in this space where you see is gray in the chloroplast. So this entire structure is a chloroplast, and inside here, this gray area will be the stroma. The light green areas will be the thylakoid membrane, where the light-dependent stage occurred. Now we are going to the light-independent stage, which will be in the gray area. All right. Now, there are several pathways to how plants go about making the actual glucose. Several pathways. However, for our syllabus, we are only focusing on the Calvin-Benson cycle. Sometimes in a book you might see the C3 cycle, and sometimes you might just see the Calvin cycle. All right? The other cycles, CAM and C4, is not really part of the C6 syllabus, which I find a bit interesting because C4 in particular is typically the pathway that most plants in Jamaica would use or in the region, because C4 are usually adapted for tropical climates. All right, some terms you are going to come across while we are doing this. RUBP. It means ribulose biphosphate. Now, RUBP can be used. You don't need to remember all this long word, but RUBP is international. You can use it. Just write it in that way that I've written it, where you have capital R, common U, capital B, capital P. Rubisco is also internationally known, so that is also next term we will be using a lot. And if you notice, it says it's ribulose biphosphate carboxylase, and those of you who have been doing biology for years should know as soon as you see carboxylase, the LASE section of this name, you should immediately know, I'm hearing my students saying it, enzyme. So you know this is an enzyme. Okay, now this is where it's going to be at. Okay, where we are going to now focus on what goes on in the light-independent stage of photosynthesis. Now, previously, when we looked at the light-dependent stage, it focused on the energy that was used was from light. This section, the energy source is the ATP and the NADPH that was made previously, along with a lot of enzyme. So it has three sections to it. The first phase is called carbon fixation. The next section is called reduction phase, and then you have the regeneration phase. Now let's go through this sequentially. One great thing about the syllabus, you do not need to know every detailed biochemical structure that is here. What we need to do is follow the number of carbons. And if you do that, you'll be great. Now, let's start. First of all, I need to bring your attention to this word, fixation. Now, in science, in biology especially, fixation has a special meaning. Okay? The word fixation has nothing to do with what you probably see in the dictionary as fixation. Fixation means, for biology, is the ability for the process by which some inorganic form of an element is incorporated into the biology or into the biota of the environment, which means into the living organism. So carbon dioxide as a substance cannot be utilized by living things, okay? It doesn't really have much use. However, with the use of this enzyme Rubisco, it helps to catalyze the reaction between carbon dioxide and RUBP and to make a compound called 3-PGA. Now, just to let you know that in this way, it means that now the carbon dioxide that was once an inorganic substance has now incorporated itself into an organic substance. That is what fixation is. This part of the process only requires enzymes, okay, for it to happen. And the enzyme is Rubisco, which is considered one of the most popular enzymes in the world because it is utilized each time this cycle goes through. All right? Now, what I need to draw your attention to is that RUBP is a 5-carbon compound. So when you add this 5-carbon compound, and obviously this is a 1-carbon compound, okay? When you add 1 to 5, mathematics will tell you, you'll end up with a 6-carbon compound, okay? Okay, I can't write some, I have the worst handwriting, people, so bear with me. So you end up with a 6-carbon compound. The problem is this 6-carbon substance that's made is not viable. It cannot stay. It is very unstable. So therefore, immediately as it's made, it's broken into 2 compounds of 3-GPA, which is a 3-carbon compound, which is a 3-carbon compound here. As you can see, so there, obviously, if you have a 6-carbon substance being made, the 3-carbon compound here, it means that there will be 2 of them being made here for each of this thing happening. So this PGA is actually phosphoglyceric acid, all right? And what happens is the next stage, when after this is made, the ATP and the NADPH from the previous stage of photosynthesis is now utilized here to convert this 3-PGA or phosphoglyceric acid into glycerate phosphate, okay, or phosphoglyceraldehyde, sorry. So it is going to convert the phosphoglyceric acid into phosphoglyceraldehyde. Now, phosphoglyceraldehyde is still a 3-carbon compound because what happened is in this conversion, all you have is what's called a reduction. Again, just like with fixation, the word reduction in science has a specific meaning to it. And that meaning is that a substance is either going to gain some electrons or the substance is going to gain hydrogen. And that is what reduction means from a chemical point of view. Not that it's reducing in size or anything. So there's no reduction in size. And that is usually a misconception that students have when they say the reduction phase, they think there is a loss of something. But that doesn't happen. What actually is happening is that the energy from the ATP is used to ensure the formation of the hydrogen that is coming off of the NADPH is added to the phosphoglyceric acid to form phosphoglyceraldehyde. That is what reduction is. All right? Now, one thing you need to also remember now is this. Phosphoglyceraldehyde, or G3P as it's called, all right, is actually the first carbohydrate made by photosynthesis. I know. In grade 10, probably even in primary school or so, you must have come up where photosynthesis is a process by which plants uses carbon dioxide and water to make glucose. Yeah, we know that. But initially what's made is phosphoglyceraldehyde, okay? And as you can see, there are still 3 carbons in this substance, and sometimes called a triose phosphate. But notice that if we had 2 of them up here, you will still have 2 of them here as well, because you still have not lost any carbons. You still have your same 6 carbons after the initial reaction between carbon dioxide and RUBP. Now, what happens is that the next stage in the cycle is where the 3GP, or the phosphoglyceraldehyde, is going to cause, there's going to be a regeneration of the RUBP because it's a cycle. So whatever we start out with, we must end back up with. That is basically what a cycle is about. So therefore, what happens is that the ATP, more ATP from the previous stage of photosynthesis is now going to be utilized to convert our G3P back to RUBP. But wait, we know that glucose needs to be made eventually. So how come that all I'm talking about is just this cycle? You're still not hearing nothing about no glucose. Hold on, let me explain. What happens is this. This process is going on in the stroma of the chloroplast. And in the stroma, it's very free. It's just a gel-like space within the chloroplast. And so this process is happening millions and millions and millions of times over and over and over again. So what happens is that every time this cycle goes on for 6 times, it has to happen 6 times. Each time, for every 6 times that this cycle goes through, you get an output of 1 glucose. Okay? For every 6 times. So just this one cycle that I have outlined to you would not be enough to make one glucose molecule. But what happens is, because this is happening several million times inside that one chloroplast, they will definitely have enough times where the each time it goes 6 times, one glucose molecule is made. Because, as you can see, if this cycle goes through 6 times, 6 carbon dioxides will be used up. And if you remember clearly from the original photosynthesis equation, you would have noticed that it starts out with 6 CO2, right? So the 6 CO2 has to go through this cycle for that eventually will lead to the one glucose molecule being made. Okay? If you remember the equation correctly. All right? Now, in most books that you are going to look at, you are going to see the diagram outlined like this, where they're going to do it where they say 3 times, they're going to show 3 RUBP reacting with 3 CO2. But it gives the impression that you need to have 3 of them for 3 of them to react, and really it's a one-on-one relationship. So what happens is it goes through the stages 6 times. I know 3 times, because if you notice, at the end they say half a glucose molecule is made, because you only went through, you only use 6 carbon dioxide molecules, all right? Oh, sorry, 3 here. So therefore, you only can make half of a glucose molecule. Remember, glucose as a molecule have 6 carbons in it, all right? So therefore, if you have 3 carbon dioxide being used up, you only can make half of a glucose molecule. And that really doesn't really exist. So what happens is that this reaction goes through several times before a glucose is made. And I know a lot of persons are saying, because my students say it all the time, so miss, this is very inefficient, because you mean that how the plant have to do this whole 6 times before they get one glucose molecule? But when you think about it, in every chloroplast that exists inside of one leaf, and each leaf has several cells that have several chloroplasts in it, hundreds of chloroplasts. So when you multiply that, it's an enormous number when you consider an entire plant, okay, going through photosynthesis. So even though it may not be efficient in our heads, it is very efficient to ensure the viability of all life on Earth. Because photosynthesis is where life on Earth, food for everybody on Earth really starts. It's from photosynthesis. So just a little recap. Carbon fixation is the first part of the process, which is CO2 combines with your RUBP with the aid of ribisco to make your phosphoglyceric acid, the PGA that was written on the other slide. Then we have reduction of the GP, or the phosphoglyceric acid. Sometimes we call it GP because depending on the book you read, you're going to see GP. Now, GP is converted to glyceraldehyde 3-phosphate, which is the first carbohydrate of photosynthesis. All right? And this process is where the ATP and the reducing power of NADPH, because remember, this is the reduction phase. So what happens here is that NADPH gives up its hydrogen to the process to make the glyceraldehyde 3-phosphate, and so therefore it becomes what's called oxidized, so it can go back to the thylakoid membrane to pick up more hydrogen ions and electrons. All right? And then you have the third phase, or part of the cycle, where it's the regeneration of RUBP, and this stage also requires ATP. Okay, so now we have done all of this. It's time for some questions. And no, I am not the one, I'm not the teacher to ask, little simple questions, but we'll try and see what we can do. All right? So here are some questions for you. First question is, we're going to start simple. Where in the chloroplast does the light-dependent stage of photosynthesis takes place, and why? So that is a question. The second question, give the absorption wavelength for the chlorophyll A at the reaction centers for each of the photosystems. And the third question is, describe where in the process are the following produced, the NADPH and the oxygen. Okay, clearly all of these questions is for the light-dependent stage of photosynthesis. Let's see. I hope you have your answers in mind. Let's see how close you came to them. All right. So the first question that asks about where does the light-dependent stage takes place, it will have it on the thylakoid membrane. This is where the photosystems and the ATPase can be found. So in CAPE, when they ask certain questions, you need to be very careful as to the verb that the question used. So not only did the question ask where did the process take place, it asks why. So it took place, or the light-dependent stage takes place on the thylakoid membrane. And the reason for that is because the photosystems that is important to the whole process, and the ATPase, that's the protein where the hydrogen ions need to leave the lumen of the thylakoid from or through, that is where these things can be found. That's why it has to happen there. The second question was very simple, and it could be a multiple choice question in real life. And what you find is that the photosystem 2, which is the first one we tend to look at, its reaction center has a chlorophyll A that absorbs at 680 nanometers. And for photosystem 1, you have 700 nanometers is their reaction center absorption wavelength. Please note, class, when you are writing numbers, when you ask for a length, any sort of number in science that requires some sort of unit, do not forget to put the unit, okay? Take it from me, someone who have marked several CXC exam papers, to know a lot of students have missed out on simple marks because in the haste of writing answers, they neglect to put the units. So don't just put 680, put 680 nanometers so we know that you know what you are talking about, okay? That's just a tip. The next question asks about where different things were made in the process. And you had the NADPH. And NADPH is made at the end of the Z scheme, okay, where the NADP plus is the final electron acceptor. That's where that would be made. The oxygen that is made from this process is from the splitting of the water molecules. That produces the 2 electrons that's needed to replace the ones that are lost from P680. Please note, and that's the next thing that students tend to forget, the water molecule splitting to produce those 2 electrons happen at photosystem 2 to replace electrons from P680, not the photosystem 1, P700, because P700 gets its electrons from the electron transport chain. All right? Here's our next question for you. The light-independent stage of photosynthesis does not require light. However, when it's dark, photosynthesis stops. Explain the following. First, how will the darkness affect the light-dependent stage of photosynthesis, and how will darkness also affect the light-independent stage of photosynthesis? Now, typically when CXC asks this question, they tend to not split it up like this. And what most children do, they answer the question and they only focus on if there is no light, there is no light-dependent stage. And everybody knows that. That is very low level, okay? However, we need to know and recognize and appreciate that the light-independent stage is affected by the presence or absence of light as well. All right, so here's the answer. I just put on an answer key, not a full thing, when you are answering the question, you'll use full sentences, nice paragraph formation, proper English and grammar and all of those things. That's expected. In this answer, you must mention for the light-dependent stage, it will stop because there will be not enough energy to excite the reaction centers. It is not important just enough to say, oh, well, there's no light, so therefore the light-dependent stage will stop because it requires light. No, that is like a grade 10 answer. At sixth form, we expect you to not only be specific, but specifically detailed. So you need to specify the fact that P680 and P700 will not get enough energy to be excited. Also, there won't be any splitting of the water, so therefore there will be no hydrogen ion for the final part. Okay? For the light-dependent stage, if there's no light, there will be no ATP or NADPH to provide the energy to convert your GP to your TP. You need to be specific and detailed. It also would be no ATP for the regeneration of the RUBP. Okay? So you need to be specific like that. All right? Last question. Now, there are other things found in the chloroplast. Your RNA, your ribosome, DNA, and ribosomes, thylakoid members, stroma. You need to discuss each of these importance to photosynthesis. And next question that could be asked on the exam. So let's go and see what we could say. First of all, the DNA and the ribosomes, they provide a platform to make the necessary proteins for the process to occur. Because the enzymes and the ATP synthase and so on, they don't come into the chloroplast by magic. You need the DNA and RNA and ribosomes to make these. Thylakoid membrane, as you know, this is where the light-dependent stage photosynthesis takes place and the production of ATP and NADPH. The stroma, this is what it does. This is where the light-independent stage takes place and ultimately the production of carbohydrates. Now, homework. So this is a question that asks about how photosynthesis can be slowed down or sped up because of temperature of the environment. And you just state specifically what stage of photosynthesis is most likely affected by the temperature changes and why. Okay, so here's the answer. Oh, no, this was homework, right? So no answer for you today. All right, so I'm going to wrap up now. I hope you remember the various stages of photosynthesis. I do have a Google Classroom that I did open for you guys. This is the code for it. So if you have access, feel free. There'll be more information there. And you could even post the answer to the homework question. There are other questions there for you to do and other material as well. So I'm asking you to please help and join the Google Classroom for this. So in recapping, 1, photosynthesis, sorry, that's a word sometimes. Photosynthesis is a process that plants undergo. It's an important process, not just for plants, but for the entire biota of the environment. When I said the biota, I mean all living things on the planet depends on photosynthesis. That's why it's such a crucial topic to know and to remember and to understand. Because the knowledge and ideas behind photosynthesis is used by many people in industry, including farmers. Okay? Because farmers who have to grow, whatever products and plants and things like that, they have to, if they have the knowledge of whether or not the amount of light plants need, when they have specialized greenhouses and so on, they can control that environment to ensure that the plants have better yield, they grow faster, and they develop better. And so therefore they end up having more substances, more fruits and for production, more for selling, more for their own families. So that is very important. And not only that, there is also scope for photosynthesis in that where it leads on to other aspects of the ecosystem, because that primary making of food is now leads to the next level of the trophic level in the ecosystem, which is the consumers. So when the consumers eat a plant, they incorporate all of that what the plant makes into their biosphere as well. Okay? Please also remember that even though glucose is primarily made, that the glucose is going to be converted to various things, including other carbohydrates, lipids, and even proteins within the plant. So therefore, photosynthesis actually is just the foundation for which all foods come from. So really and truly, today, I hope that I have enlightened all about this particular topic. And I do know that some students in class tend to have issues with this topic, and I want you to really take note and heed of how the process has been deconstructed at grade 13. So that's all the time we have for Cape Biology.